think I already mentioned your facilitator this evening. So today I have with me Christopher Saunders from the Pirbright Institute and Jose Bis from the APHA, and we will be talking about Bluetongue for veterinary professionals. You will all be on mute throughout this webinar, and any questions, if you have them, please put it in the Q&A at the top. So hit that Q&A button. There is, I think, a chat button, but that one will not work. So just put your questions there. And at the end of the session, so at the end of both the presentations, we can discuss them. And please do use this time and do ask loads of questions because I'm sure you'll have them. Just so you're aware, the webinar will be recorded, and this will be posted on the APHA YouTube channel, and that will come out in due course. When it's out there, we will send you a link to those that have registered to this webinar, so you can also look back there, and there'll be a link on the gov.uk website as well. So what this webinar is obviously aimed at veterinary professionals, so be aware of that, just in case you've dialed in and you're not a vet. Just to let you know, we'll be talking about the transmission cycle, the pathology and the pathogenesis of Bluetongue, clinical signs and differential diagnosis. And with that as well, clinical signs that were seen in Holland last year or over their Bluetongue season, as well as control and prevention. With this, I'd like to hand over to Chris. Oh, sorry. So there are some links, but you'll get them. So I'd like to hand over to Chris for his presentation.
Okay, thank you, Sasha. Just checking you can hear me before I start. Is that okay? Can you give me a thumbs up?
Yeah, I can
thank you very much.
Hear you. Perfect.
Lovely. Thank you and welcome, everyone. So my name is Chris Saunders. I'm a research fellow at the Pirbright Institute, and I've been working on Bluetongue virus and the midges that transmit Bluetongue virus since 2006. So today, as Sasha said, we're going to talk about the transmission of Bluetongue virus by midges and the clinical signs we can expect to see resulting from Bluetongue virus infection. Next slide, please. So just as a reminder, hopefully you're aware of Bluetongue virus. Bluetongue virus is a non-contagious vector-borne virus, and it's the causative agent of Bluetongue disease, or BT or BTD, of ruminants. So sheep, cattle, deer, goats, and also camelids. Bluetongue virus is exotic to the UK. We want to keep it that way. And when we talk about Bluetongue virus, we're actually discussing quite a complex situation. There are more than 29 different serotypes of Bluetongue virus and many different strains per serotype. So effectively, we have more than 29 different diseases, and each of these strains or serotypes can have different characteristics in terms of the disease they cause and their transmission. We've seen dramatic changes in the epidemiology of Bluetongue virus since 1998 in particular. If we've been talking then, we would describe Bluetongue virus as a tropical or subtropical disease. But since then, we've seen the emergence of exotic viruses into the northern temperate regions, including the UK. But we've also seen an increase in global disease incidence and an increase in virus diversity in endemic regions. Next slide, please. So these are the problem insects. I am an entomologist, so this is my main research area. Bluetongue virus is transmitted by Culicordes biting midges. These are some of the smallest blood-feeding insects. They can be 2 to 4 millimeters long, so much smaller than a mosquito. They have a near worldwide distribution of about 1,500 species worldwide, including 50 in the UK. We can tell them apart because they have these quite pretty patterned wings, as you can see on the bottom right there. And one thing that's typical of Culicordes is they're hugely abundant. So we can trap hundreds or tens or hundreds of thousands of insects in a trap in a single night, and not really dent the adult population. They're typically crepuscular, so they're active at dusk and dawn, a little bit more overnight. And unlike mosquitoes, they don't breed in free water. They breed in damp organic matter. So that includes soil, leaf litter, and animal dung, essentially the farm habitat A few species worldwide are vectors of Bluetongue virus, including some UK species, and it's the requirement of females to blood feed on vertebrate hosts to mature their eggs that leads to their role as vectors. Next slide, please. So this is the transmission cycle of Bluetongue virus. We start with our infective host on the left, this animal that has a viremia. In comes an uninfected adult vector. It bites the ruminant and acquires virus within that blood meal. That virus then has to travel from the gut to throughout the midge to the salivary glands, and this is called the extrinsic incubation period, which we'll talk about in a minute. Once that period is complete, the infected adult vector can bite and infect a susceptible ruminant host, and that susceptible host then, after a latent period, develops viremia and becomes infectious to vectors. Now, it might be strange for me to say that actually culicoides aren't very good at acquiring Bluetongue virus. I've just said to you they're the vectors of Bluetongue, but there are lots of barriers to infection, and actually they're not very good at picking up the virus. But what they are incredibly good at is passing it on. So once they become infected, that efficiency of transfer from an infected midge to a susceptible host is very high. In fact, one bite from one infected culicoides to a susceptible host can initiate the full viremia and clinical disease of Bluetongue. Next slide, please. So culicoides are what we call biological vectors of Bluetongue virus. So to be transmitted by a midge, Bluetongue must replicate, and it must disseminate from the gut through the body of the midge, from the blood meal, eventually getting to the salivary glands. And you can see the salivary glands in red and green on that top confocal image. Now, I said they're not very efficient at this, and it's only possible in a small proportion of individuals, even of vector species. And we call this proportion that's able to transmit the virus the vector competence of that species. And this is typically from 1 in 1,000 individuals to about 15% of individuals that feed can actually then transmit the virus. The extrinsic incubation period is the time taken from the virus to get from that blood meal that's been acquired to get all the way through the midge systems to the salivary glands, from when it can be inoculated into a host when the insect next bites. And the time it takes is dependent on both the viral strain, the vector, and also ambient temperature. Next slide, please. So because these insects are exposed to environmental temperature, all their bodily processes are also dependent on temperature as well, but the duration of EIP is directly related to temperature. This is a function of virus replication, and because it's in the midge for at least part of its life cycle, it is subject to ambient temperature. Bluetongue replication can only occur when the temperature is over 12 degrees centigrade. As the temperature increases, that replication speed increases, and so the duration of the EIP shrinks. In addition to the EIP, then, the viral dynamics, the activity, survival, even the development of midges is also dependent on temperature. So although midges in the UK are active above sort of 4 degrees C, they're most active at temperatures of 18 to 20, maybe 22, up to 24 degrees C. And so high temperature and in the environment means high activity of culicoides. They're biting more, they're producing more offspring. We also get high virus replication in those midges. And so if we get high temperatures, we're more likely to see increased Bluetongue virus transmission, and therefore temperature is a real significant driver of Bluetongue virus transmission. Next slide, please. Okay, so what can we expect when we see Bluetongue virus in animals? Well, Bluetongue virus can affect all ruminants. The clinical severity of disease is very variable. It depends on the virus strain, the species of animal, of ruminant, the breed of that ruminant, and there's substantial variation between individual animals. Severe clinical disease is typically only seen in sheep, particularly the fine wool breeds of improved sheep. Cattle, typically are subclinically affected. So although they're infected, they don't show any external signs of infection or disease. But they're really important as they act as reservoir hosts. Next slide, please. So what do I mean by reservoir hosts? Well, we actually get the same sort of pattern of infection in both sheep and cattle. So although there's a difference in the disease outcome, the actual virus replication is pretty similar between the two species. Now, one of the interesting things about Bluetongue virus is that the Bluetongue genome is actually detectable by BCR for a very long period post-infection. So we get a peak of viremia sometime between 5 to 10 days post-infection, and this is the most infectious period to midges. But after this, we start getting antibody production, and we enter what we term a plateau phase. So sometime after 10 days post-infection, we're still getting a PCR signal, these animals. But because the antibodies are active, our ability to isolate virus becomes increasingly harder, particularly after 20 days post-infection. And we think that the infection of midges also follows that pattern. So the most infectious period is at peak viremia. Next slide, please. So clinical disease of Bluetongue in sheep, as I mentioned, is dependent on the breed of the sheep. It's also dependent on the age of the sheep, the strain, serotype of the virus, and naivety of the population. And even in sheep which we consider susceptible, this disease can range from mild, moderate to severe. So Bluetongue 8, we had about a 30% case fatality rate, and that was considered a pretty severe strain, or a damaging strain. So the typical signs of Bluetongue virus, or Bluetongue disease, in sheep develop somewhere between 5 to 14 days post-infection. And typical signs are a fever and reduced appetite, lethargy, and depression. You can see in the image on the right there, the ears have gone down on that animal, its head's gone down, and it does look depressed. And that's what we mean by depression. We also see reddening of the conjunctiva and increased tear fluid. We see hyperemia of the nasal and oral mucosa. They go very pink. We see edema of the lips and erosive lesions of the lips and oral mucosa. We quite often get facial edema. So these animals look like they've got fat faces, essentially. And this we sometimes get clear and stringy, but not frothy salivation, and a mucopurulent nasal discharge. Sometimes these animals go into respiratory distress, and they can have a stiff or uncomfortable gait or associated lameness. This may be partly due to reddening and swelling around the coronary band, which is painful on palpation, and in severe cases can bleed. Generally, we see a decreased production, including weight loss and wool breakage from severely affected individuals, and there's also reduced fertility and fertility issues, including abortions and weak lambs. So Bluetongue virus Bluetongue disease is typical hemorrhagic fever from what the virus is actually doing to those cells. So infection of the endothelial cells of the blood vessels increases vascular permeability. Essentially, the blood vessels start leaking. So serum and lymphatic fluid leaking into tissues causes edema. When the endothelium of capillaries is severely damaged, we get petechial and hemorrhagic bleeding, and leading to tissue necrosis as that oxygen exchange is prevented. With the inflammation this causes, we see increased blood flow, the hyperemia I mentioned. We get more inflammatory cells, making the problem worse, in order to clear those proteins. But that results in the cardinal signs of redness, heat, swelling, and pain. This tissue damage has a pyretic effect, causing the fever that we see. And the movement of affected tissues, particularly the lips, the nares, dental pads, anywhere where there's kind of movement, can lead to erosive lesions caused by this mechanical damage due to sensitive tissues. We do see mortality associated with Bluetongue disease. This could be a result of severe lung damage that's causing asphyxia. We can see disseminative intravascular coagulation, or DIC, so blood clotting in these animals, as the blood clotting processes break down due to infection. Secondary infections, particularly bacterial pneumonia, are really important cause of mortality associated with Bluetongue. And anorexia resulting from an inability to eat because their mouth is sore, or swallow properly because their face and throat is swollen with edema, or if they're too lame to walk to find food. We get these animals that just aren't doing very well. So some pictures of clinical signs of BTV in sheep. This facial edema, you can see the puffiness around the face, particularly the lips. We see this nasal ulcers and excess salivation. And again, notice that the ears of that animal are down. We see that quite often as an early indicator of the sick animal. Next slide, please. This is the inflammation of the coronary band. So we can see swelling and redness, but also sometimes ulcers, and that can be really quite painful. We sometimes see redness between the digits as well. And then when we get to PM, we can see hemorrhage in all the mucosal tissues, particularly in the oral mucosa in the mouth, the dental pads, as you can see on the top left there, and also subcutaneous hemorrhage, both just underneath the skin, but also over the top of the muscles as well. Pulmonary edema and lung damage due to congestion results in asphyxia, and that's you can see the throbbing in the trachea at the bottom of the extensive damage in that set of sheep lungs at the bottom. Next slide, please. So I mentioned oral lesions. These can be quite extensive and quite sore for the animal, so it can stop them eating. You also see hemorrhage, particularly under the gums and sublingually. You can see on the bottom left, there's that petechial hemorrhage underneath the tongue in that animal. Next slide, please. In cattle, we typically don't see clinical signs of bluetongue. And we know they can be affected because we get the PCR results and things like that. But if we do see clinical signs, they're typically mild, but like those seen in sheep. We can see a rise in body temperature and fever, the reddening of mucosal membranes, including conjunctiva of the eye and udders. We can see subcutaneous edema, swelling of the face and lips, as we saw with sheep, and similar ulcers and petechial bleeding on the nose, gum, and dental pads. The coronary bands are also somewhere we might see inflammation and reddening, and that might be associated with lameness as well. Again, nasal discharge, excess salivation, and the behavioral changes, which are perhaps our first indicator that something's not right. And we can also see a loss of appetite. Next slide, please. So these are just images from FLI in Germany, excuse me, of the nasal reddening and crusting of the nares that we see in the few cases of BTV in cattle. Redness, inflammation of the udders and teats, and we can sometimes see a reduced production in cattle, particularly in reduced milk production, and also infertility problems as well, including abortion and transplacental infection. So I mentioned the kind of reduced fertility and abortion, but we also get sort of mummification and this the development of dummy calves that are essentially born with hydroencephaly, substantial parts of their brain missing, and obviously these calves are non-viable. So there's evidence of Bluetongue virus transmission in pregnant ruminants both in the UK and the EU. So hydroencephaly is reported, and there are some reports of arthrogryphosis in newborn calves as well. But the effect that we see is dependent both on the stage of pregnancy at which the infection occurs and the severity of the disease as well. Next slide, please. So if we see infection in the first trimester of pregnancy, we might see fertility problems and abortion, leading to teratogenic effects, disfigured calves heading into the second trimester. And then by the middle or end of the second trimester, we're seeing infected calves born with developing an immune response. The clinically ill or completely normal calves born, but if the calves survive, the infection will be cleared. There's also a interaction with colostrum of maternal antibodies given to these individuals as well, which might cause the infection to be cleared much quicker. Next slide, please. So hopefully you're aware that Bluetongue virus 3, serotype 3, was detected in the Netherlands in September last year. And there was a substantial number of infections and severe disease was reported. Bluetongue virus infection was identified at more than 4,000 locations, including 1,900 sheep farms, 2,200 cattle farms, with a further 1,500 farms with clinical signs, but with no confirmation of Bluetongue virus infection. Close to 5% of the sheep population has died, according to the excess death figures, And they did actually see a bit of mortality in the cattle population as well, but most markedly was the milk production losses across the national herd. There's a suggestion that BTV3 in the Netherlands spread faster and had more severe morbidity and mortality than seen with BTV8. And as we get the confirmation of this data, once we understand fully what's going on, we'll be able to compare them more. So next slide, please. So hopefully you can see that the video, it's not playing particularly well on my screen. Thanks to René van der Brom from Royal GD in the Netherlands and colleagues at Wageningen for these pictures of what they saw in the field in Netherlands. So in sheep, they saw apathy, isolation, lameness, and excessive salivation, as you can see from the still of the sheep's face. Erosion and ulceration of the mucosa, particularly in the mouth, pulmonary edema, hydrothorax, and hydropericardium. Associated with a lot of these cases, they saw extensive organ damage. And even when these animals survived the acute phase, if they did survive the acute phase, it was also linked to periarthritis, evidence of muscle necrosis, and also deshoring where the hoof was shed. We don't know the extent of the mild or subclinical cases in this background of the extent of severe disease as yet, and so that's kind of part of the picture that's still missing. In cattle, they reported much milder disease than in sheep, but also saw reported disease in goats and alpaca. But acute disease and death was also reported for cattle. Again, they saw the fever, the significant drop in production, particularly milk production, nasal, oral, and udder lesions, nasal discharge, conjunctivitis, excessive salivation, lameness, stiffness, and swollen coronary bands. You can see from the picture on the right there, a very sore-looking hoof with inflammation of the coronary band. Deshoring is also reported. Oops, sorry, you can go back. So deshoring is also reported in some of these animals, and regurgitation associated with the swelling and the damage done to the throat. Next slide, thank you. So, in summary of the current situation in the UK, so Bluetongue virus 3 was first detected in November last year. The virus is the same one as was causing the outbreak in the Netherlands. Subsequently, Bluetongue virus was confirmed in 126 animals, including 7 sheep across Kent and Norfolk. All the animals were PCR and seropositive, so they had virus genome and also antibodies to Bluetongue virus infection, suggesting they were quite old. So they were older infections that the infection had happened earlier. No clinical disease cases were confirmed in the UK. But we did manage to isolate a virus from a UK animal, Pirbright, and so we underwent an experimental infection of UK sheep under standardized conditions to really characterize the disease in UK sheep. Next slide, please. So we used our infection model under standardized conditions. We use a midge-borne infection system, so we actually infect midges first and then use them to infect the animals. And then we can examine them closely to understand the clinical signs, the pathology, and the diagnostics associated with that infection, and also look at transmission, onwards transmission to vector-culicoides. Next slide, please. So for this study, we used North Country mules, which Swaledale and Leicester crosses. We did the study in February, and it's actually quite difficult to get sheep that weren't in lamb. But we got six mature ewes of about 6 years old or more. We infected culicoides and fed them on 5 of those sheep and left 1 as an uninfected control. All 5 sheep received bites from the infected midges. And then we could take the midges back to the lab and confirm that they were actually infected once they're fed. And so we got somewhere between 16 and 29 midges feeding on each of the sheep that we had. Next slide, please. So in summary, all 5 sheep exposed to bites became infected with BTV3. And you can see the PCR results on the right. All of them have a similar viremia. 1 sheep seemed to come up much quicker and was positive by day 2, and into day 3 was reaching peak viremia. One thing we saw was a substantial individual variation in disease. 3 out of the 5 sheep, you can see that their lines stop on the graph. 3 out of 5 sheep were euthanized at days 10 or 12 days post-infection, having reached the moderate humane endpoints of the study. One sheep, in fact, the sheep 2 that came up really quickly, recovered from moderate disease following treatment with non-steroidals. Despite having very high temperature, it did seem to recover. And one sheep exhibited very few clinical signs. It showed a bit of a peak in temperature, some redness of the eyes, and then continued as if nothing had happened. And our control animal, fortunately, remained uninfected and unaffected. Next slide, please. So what we saw in this sheep was in all 5 sheep, we saw a high fever. The duration of that varied between animals. Some animals seeing a fever for more than 4 days. In 2 animals, we saw hemorrhagic diarrhea, and this was the humane endpoint for this study. So unfortunately, they had to be euthanized at that point. You can see on the right the sheep that refused to move and get up. So this animal, even through examination, kind of refused to stand, refused to get up, refused to move, and was obviously very sick. So that animal had to be euthanized at that day. We also saw some facial edema. This particular breed of sheep does not show a substantial amount of facial edema from our previous studies, but we did see some, and reddening of the ocular mucosa and oral mucosa. Particularly in one sheep, we saw a particular reddening and swelling of the coronary bands, although all infected sheep showed some degree of reddening. One sheep looked like it had pink socks on, where that reddening was quite apparent, and those feet became quite sore. We did have one sheep that developed an oral lesion, and also we saw nasal discharge. Next slide, please. When we took these animals to PM, we saw a petechial hemorrhage, particularly sublingually, so under the tongue there, you can see in the top left picture, but also in the stomachs and lymph nodes, particularly in the junctions between stomachs where there's a lot of mechanical movement as the animal digests. In one animal in particular, the one that wouldn't get up, we saw a lot of subcutaneous edema and edema within its joints as well. And you can see that yellow buildup of fluid in the bottom left picture. We also saw hydropericardium, so fluid around the heart, and substantial ascites with more than 30 mils of excess body fluid in the body cavity of one of the individuals. One of the individuals' sheep also had congestion of the lung, and you can just see that the tip of one of the lobes there is congested as well. And that was the most sick sheep that we saw. Next slide, please. So the clinical picture for that we saw suggests that the Bluetongue serotype 3 will impact UK sheep if it comes back. So what can we do about Bluetongue? Well, vaccines are the best tool to use against Bluetongue virus. So vaccination of the animals, the success of the vaccination campaign against BTV8 in 2000 meant that we didn't actually see any infections in the UK in 2008. This is in stark contrast to the continent, particularly France and Germany, where they weren't able to get that vaccine rollout in time, and they saw substantial damage to their livestock industry. Unfortunately, the vaccines that work really well are serotype specific with very little cross-reactivity and cross-protectivity. So a BTV8 vaccine will not protect against BTV3. There is no vaccine for BTV3 currently available, which leaves us in a bit of a quandary of how do we advise people on what to do. Next slide, please. So one thing that's been suggested is that we can break that transmission cycle by controlling the vectors. And there's been a number of suggestions made to how we could go about this, and none of them have been shown to be particularly efficacious in the field. If we remove dung, suggesting that this is a large larval habitat for these insects, they do certainly breed in dung habitats, but removal of dung heaps has not been shown to affect Culicoides populations substantially because they are capable of breeding in many diffuse habitats within the farm environment. Similarly, insecticidal spraying of vector habitats is not recommended because you just cannot target everything to be effective, and then there's environmental concerns associated with that. Repellents that would prevent contact with the animals, basically shoo the culicoides away from the animal, are problematic because they're so transient in effect, have not been shown to affect Bluetongue virus transmission in the field, and have not been shown to reduce biting rates substantially on animals. Poor on insecticides such as synthetic pyrethroids may kill culicoides in the lab, but again have not been shown to affect Bluetongue virus transmission in the field. Again, the persistence lasts days rather than hours, but it's still going to have to be a repeated treatment. And timing of that treatment to prevent onwards transmission is going to be really, really difficult. So we can't recommend synthetic pyrethroids as a method for controlling Bluetongue transmission. Not been shown in the field. Stabling of animals, bring them into stables to protect them, has been suggested. Unfortunately, if you bring animals, it's very context dependent. If you bring animals into a shed, you are essentially concentrating the smell of those animals that these insects use to find their hosts. So concentrating them may not actually be beneficial. Culicoides still enter stables and sheds. If you've got a kind of varying degrees of animal sheds, if it's got an open side or open doors, they're still going to enter, particularly when it's inclement outside in the autumn. We actually find more midges inside cattle holdings in the autumn than we do outside. Vector proofing accommodation is possible, but this comes with a caveat. You need screening with mesh of sufficiently small aperture that midges can't get in, and this means less than 0.5 millimeters in size, which is actually difficult to get hold of commercially. You've also got the issue of ensuring airflow to the animals to prevent kind of other health impacts associated with essentially boxing your animals in, and the duration of that closure may also be problematic. The Dutch have given us anecdotal evidence of an impact of increasing ventilation within an animal holding that had been screened. So essentially using positive air pressure to push culicoides out of that environment. But the logistics of that are going to be very difficult to work out on farm. Next slide, please. So in summary, Bluetongue virus is a notifiable disease, and it affects all ruminants and can cause disease in sheep and sometimes cattle. Bluetongue virus is spread by culicoides biting midges, and that's why it's seasonal and why it's so dependent on environmental temperature. Clinical disease associated with Bluetongue virus and Bluetongue disease is highly variable and depends not only on the virus, but also on the host species, whether it's cattle or sheep, and also the breed and age of those animals as well. And even within that, we have substantial individual variation. Bluetongue serotype 3 could overwinter in Europe. It could persist in the UK or arrive in the UK this summer. And it certainly could impact ruminant health and welfare and trade of livestock in the UK quite substantially. There is currently no vaccine for Bluetongue virus 3 licensed for use in the UK. And unfortunately, there are limited options for on-farm mitigation in terms of vector control that would impact Bluetongue virus transmission. Thank you very much. That's me.
Thank you so much, Christopher. That was really interesting. And I must say, it was also really interesting to see the research that Pirbright did, and that all those sheep that actually were infected did show some point clinical disease. So thank you so much for sharing that. It was, yeah, I'm sure there'll be questions, and please, I just want to remind people, if you have any questions to ask, Chris is here with us now, so do stick something in so we've got something to talk about at the end. So I'd like to now introduce Jose. So Jose will take on the next part about disease and control and prevention. Thanks, Jose.
Thank you, Sasha. Good evening, everybody. Yeah, I'm Jose Bis, veterinary head, field delivery in APHA. I'm going to follow up Chris's presentation. Mine's going to be a little bit around rehearsing some of the elements that Chris has done on what was found in the Netherlands, but with a bit of an emphasis on what sort of effects that had on farmers and the veterinary professionals, the sort of things they did, and then talk a little bit about what next for the UK, what might happen. It's, excuse me if it's going to be a little bit loose, because we don't know exactly what's going to happen or even exactly how we would react to it, but we know some principles which I'm going to talk about in terms of control and prevention. So next slide, please. This first one is just a pictorial map of the Netherlands. So can you go back? Sorry. So just a representation of what it looked like, widespread infection, and the numbers there of clinically confirmed cases in the Netherlands, and the number of positive PCR samples that they had. Next one, please. So Chris has talked about the effects of BTV3, in particular the serotype 3 in the Netherlands, and how it created some serious illness, particularly in sheep, but also some lost production in cattle, and some milder disease. This caused a significant amount of distress on farmers. There was a big demand for therapeutic options from their private vets, and it caused mental health problems for farmers. This talks about the anxiety of prior to visiting animals in the morning to see what was there, and knowing that there was very limited things that they could do to prevent the disease on their animals. At the end of the chat, there will be some links which we can, I think Sasha's going to put up some links that provide farming support groups that people that you can certainly advertise to your to farmers, and the farming crisis network is clearly one of them. And APHA do work quite closely with them, and they provide not just financial but also emotional support to farmers. There is this thing as well where seemingly some of the animals got better, as Chris talked before. Some of the animals did recover, but subsequently developed other health problems through sepsis, and that caused further So clinical signs in the Netherlands in cattle, it's been talked about. Just to bear in mind, most of them were mild, but there was significant production losses in milk yield, and particularly as much as 30% has been mentioned in individual sick animals. Next one, please. For sheep, is much more severe. There's this mention about face, feet, and fever. Pretty much what Chris has been talking about. But there was all these secondary elements, particularly about the necrosis in parts of the dental pad, where these lesions were subsequently colonized by maggots, causing further issues. Muscle degeneration, animals not being able to eat, and in the very severe cases, as we mentioned, dehooving and degloving of hooves. Next one, please. So this is sort of the kinds of actions that veterinary surgeons took in there. So steroids, first thing to mention. Obviously, there's risks with pregnant animals, but it certainly was effective in mitigating the generic inflammation produced by the virus and the vasculitis. The sheep took a few days to show severe dyspnea. And the recommendation was to use high initial dose of steroids or short-acting steroids, followed by longer use of long-acting types. Next one, please. So other anti-inflammatories, non-steroidal anti-inflammatory drugs, which have issues with kidney function, but certainly was kidney infection and kidney problems. So it's to be used with caution, but it seemed to be probably helpful in the acute phases of the viremia. Just worth mentioning that in the Netherlands, some of the vets used paracetamol to combat pain as well. But I'll talk later about the cascade. There's a slide that talks about what and how veterinary professionals can use in the event that they don't have a drug suitable to treat the condition of the animal. Next one, please. Some other drugs, diuretics, could be quite useful, but again, in terms of long edema that's been mentioned, but dehydration also a problem. So there's a balance there to be had. Antihistamines, no particular evidence that mast cell increase due to virus, but probably helpful as well in reducing damage due to cytokine storms. Next one, please. Yeah, antibiotics. We mentioned the fact that there was several cases of secondary infections that did occur, imbalances in the rumen and so on, leading to sepsis, and they manifested as arthritic lesions, purulent, but no specific pathogen detected. The use of narrow scope procaine penicillin or more oxytetracyclines for a wider range had been mentioned. And again, being aware of nephrotoxic drugs, given the probably the compromise of the kidney. Next one, please. This is what I was talking about. So where there's no suitable veterinary medicines authorized in the UK for the specific condition of the animal being treated, and in order to avoid unacceptable suffering, veterinary professionals can use their clinical judgment to treat animals under the care in accordance with a cascade. And you've got the link there, which will be circulated after the chat as well. And it addresses elements of withdrawal period implications in food producing species, which is also important to bear in mind. Next one, please. And so, yes, welfare is the name of the game, and euthanasia may be unavoidable. It isn't something that will necessarily be undertaken by private vets, but also the farmers may need to be well prepared for a situation where they may need to dispatch animals on farm to prevent unnecessary suffering. And there is a need for prognosis indicators, clearly, and I believe that in the Netherlands they're trying to put something together, so we may be able to get something that explains a little bit what can be expected in terms of recovering and improvement of some of these very sick animals. Next one, please. Next one. Yeah. Chris has mentioned it in much more detail. There is no vaccine for VTB3, and there are limited benefits of housing, and well, there could be some benefits of doing so at dusk, but yeah, nothing tangible that the farmers can do to prevent transmission of disease through because there's no real effective vector control method. Next one, please. And so, yeah, Bluetongue is a notifiable disease, and there are a number of, this is an attempt to put quite a few differential diagnostic diseases, some of which are notifiable and very concerning, foot and mouth being the main one, but also episodic hemorrhagic disease, which is now in France and other parts of the continent. You can pretty much think about MCF, IBR, and BVD as the differentials for cattle, with BVD also being important in terms of any sort of abortions or fetal malformations. And for sheep and goat, I think ORF is the main one. We've had a number of reports of suspect BTV3 where ORF has been found, and it can cause with similar clinical signs, particularly around crusting and pain and fever. There are a number of parasitic imoncosis and liver fluke, which can also show some of the clinical signs of Bluetongue initially. Next one, please. Oh, that's going to be a bit wonky. This was supposed to show, it was a very good representation of report case cycle, but it's gone, it's all disappeared. You've got the fireworks in the middle, but the rest of it has disappeared. Oh, it's appearing. Shh. It's slowly showing up. Oh, there we go. So yeah, typically it's back. Yes. So typically the farmer or indeed yourselves, private vets, will report to APHA suspicion of disease when you know, it's Bluetongue being in the differential. We will aim to visit those farms. Certainly, initially, we will and place formal restrictions on the farm and investigate. It will be in liaison with our colleagues from the Vet Exotics Notifiable Disease Unit, and a decision will be made whether these can be or not ruled out. And if samples are taken, the results of that will then sort of very much indicate whether disease is present or not. And following confirmation, should this happen, then there is a number of actions that will be taken, but it will be very much dependent on you know, what stage of the epidemic we're talking about. But you know, certainly on confirmation of Bluetongue in the country, there would be some type of zones put in place and therefore movement controls and some tracings quite likely. Next one, please. Next one. So I think Chris has talked about this as well. It's the current situation in the UK. I think the main thing to highlight is that there are still some risk animals which are under restrictions in the UK as a result of what went on in 2023. Those are entire males and pregnant females. And that although there is no evidence of disease circulating in UK midge population, we are back out of seasonal vector load period, as we've been able to detect, I think it's more than 5 pigmented midges in the traps that we have in the country, and therefore, yeah, we are in that period, but like Chris said, temperatures I don't think are consistently between 12 and 15 degrees, which are necessary for the capacitation of the infected midges, and therefore the risk of transmission hasn't substantially changed. Next one, please. And of course, I just failed to mention that there are currently no longer any zones in place in the UK. And so just to talk about the annual active survey, which APHA conducts. It's a targeted survey that is undertaken annually at the end of the active vector season, so kind of October, November, and it or has been, but it would be depending on when we determine that the activity of the midges has gone low. It follows a triage system where the types of farms that are wanted are selected, and it is on a voluntary basis in principle, as you know, disease is present on the country, and the legislation doesn't allow us to enforce that. It's usually undertaken by veterinary delivery partners, and it's paired up usually with TB testing in cattle farms, certainly, and it's those farms are selected in the higher risk incursion areas using meteorological information. Next one, please. And so this is how we found it in the UK last year. Chris mentioned there has been no clinical signs. We haven't seen disease in this country yet from BTV3, but we did detect it in one cow initially in mid-November last year as a result of this annual survey. And a temporary control zone of 10 kilometers was initially put in place. That was then further extended when new emerging cases were found in the northeast of Kent. Next one, please. And jump 2, so I can go back. So, and then yes, subsequently the other map is just to show that yes, the so-called Norfolk TCZ was then put in place when it was detected through the survey as well in 2 cattle in the Norfolk area, although that 10-kilometer radius also got included some small part of Suffolk. Next one, please. Next one, please. Yep. So this is just to reiterate. Yeah, the government has obviously the intention to control the spread of notifiable disease, and you know, short-term objective is to establish whether or not the virus is circulating in the midge population, and whether there's any undisclosed disease, and then of course once disease suspected or indeed confirmed, to have a swift response to effectively control and try to mitigate the spread of disease, and this might take different actions depending on at what stage of the epidemic. We will conduct obviously epi investigations on these cases to try and establish the certainly at the beginning of the epidemic, to try and establish the likely source of infection, and as it's been rehearsed already, yeah, the current hypothesis that we did get it in 2023 as a result of most likely midges blown in from the continent, knowing that, you know, in September and October last year, the Netherlands had a significant number of cases, but also in Germany and Belgium. Next one, please. And so what happened when and how we did it? these particular actions at the time. Disease was not circulating in the UK, and so we put those control zones in place. We did not find any clinical signs, and we selected, it was still a significant amount of work for the agency. We were supported by veterinary delivery partners in delivering that. We selected larger farms as a priority, try and target larger farms first. All visits included inspection, sampling of all susceptible animals, and plus record checks, not exclusively, but checking production and mortalities. We took over 48,000 samples in between both TCZs. And I think one of the issues we found, or one of the most challenging things we found, is that unfortunately, quite a number of keepers had not registered land and buildings that they were using. And so that made it very difficult to really locate all susceptible animals within those premises. And we do strongly encourage livestock keepers to correctly register any temporary use of land and buildings, and that's the link that they can use there. We'll provide them guidance as to how to do that. Next one, please. And so if we were to get Bluetongue in the UK, there's a number of, as I said, measures that might be put in place, and control zones is certainly one of them. I talked about a temporary control zone that we put last year, but dependent on where the virus is circulating and the number of infections and so on, things may be different this time around. The Bluetongue legislation allows for a number of different zones to be put in place, and these will depend very much on the evolving situation. As I said, control zone is perhaps the smallest but most restrictive in terms of movements. You then have the protection zone, which is usually at least 100 kilometers, and the surveillance zone would come at an extra 50 kilometers on top of that, but could be certainly larger. Next one, please. This just quickly maps to show, I think the green one in the middle is the sheep density maps that we have in the UK. So you can see that Norfolk and the sort of southeast counties are sort of quite devoid of many sheep. Certainly not a lot of sheep in those counties, although in Kent you've got a little bit in West Sussex. And then the blue one represents the cattle densities. Again, a little bit more cattle, perhaps in Norfolk than sheep, but not a great deal. And this map shows as well to the right, to my right, in fact, shows the at-risk counties of incursion, with certainly the east coast and the south and east coast of Kent and West Sussex being the time and time again where it's certainly where we found it. We found BTV8 in 2007 and BTV3 in 2023. And yeah, as wind blows across from the continent, those are the most likely counties to get it. So that's what we're likely to see if we see disease in the first instance. Next one, please. And so really important, I guess, at this stage to mention the necessity of, given the uncertainty, but Chris said we are likely to see something come back in the UK, probably not overwintering, but we don't know. We certainly can expect at some point, and depending on what happens in the continent, that the vector might be blown again. So infected midges might come visit again. Farmers need to be aware that they could well be caught in restriction zones. Some of these restrictions may impact them very severely, including not being able to move animals to slaughter. even on the days that they had originally planned. So they need to know that yes, we will try and expedite movements where movements do not bring further risk, but that might require abattoir designation, movement of young stock back from other premises may not be possible, particularly if it's from zones into free areas, and so yeah, they do need to think very carefully about what sort of coping strategies they can have, and there will certainly be bans on the movement of animals, and germplasm this time is, which wasn't last year, I believe, but this time could certainly be affected. Next one, please. And so talk about restrictions in place. So just as of today, APHA has launched the Animal Disease Licensing Service, which integrates the previous animal avian influenza licensing service, and it now will allow farmers to ask for licenses for not just avian influenza zones, if there were to be any, but also it integrates the possibility of those for Bluetongue zones. And what this does is, I've seen a rehearsal of this system, and it looks really good. It will allow APHA to process way more licenses much more effectively and efficiently, and it will give farmers a clear view and follow-up of where their multiple requests may be at. Next one, please. And so just to finish, yes, please do report it. We want to know. It's not just Bluetongue. Obviously, as I said before, that there are other diseases, notifiable diseases that could mimic this one. And this is the number. This is why we want to hear about it. Next one, please. Just to say thanks to a few people that have helped contribute to this presentation. And thank you all very much.
Thank you, Jose. Again, another interesting presentation, and also sounding quite worrying what happened in Holland as well, clinical pictures that was seen there. And just coming, you did mention the mental issue, or the impact that this disease has on everyone, not just farmers, vets as well. And so there are two support links, Mind Matters, that's part of the RCVS, and obviously FCN for the farming community. So yeah, please, I think what was very clear, what I understood from Holland as well, is that speaking about it and speaking to others who are in the same situation, that's really important. But let's hope we don't get that much disease here. You never know. We might be lucky. But thank you again. So I think what we'll do now is just go to Q&A. So Chris, if you're there, I'll have a look at what's been coming in. So there's a question here, Chris, about kidney pathology. Kidney pathology was seen in infected, diseased sheep in the Netherlands and given as a reason not to use NSAIDs as treatment. Was any kidney pathology seen in the experimentally infected sheep here in England?
Yeah, thank you for the question. It's a really interesting one. Excuse me. We didn't see any gross pathology of the kidneys of these animals. There was only 5, but we have yet to go through our histology samples, including the kidney and all the other organs. So we may not have had these animals long enough to see gross signs in the whole organ, but we might be able to see stuff at histological level.
Thank you for that. Another question, this is more about deer. So what are the clinical implications for other susceptible species, for example, deer and camelids?
Yeah, so this is, again, a really interesting question. Obviously, for certain farms, this is going to be really important. We don't typically see Bluetongue virus, or the viruses that have come into Northern Europe haven't actually caused much disease in deer, but they do get infected. So it would present a similar clinical picture if they were to show clinical signs, as would alpacas and the other camelids. So we wouldn't expect to see it, but it's definitely something that should be reported if you see something similar to the signs I've described for sheep and cattle in deer or camelids. It could well be Bluetongue. Just that this virus might behave slightly differently to ones we've seen previously.
Thank you for that. Well, you're going to be a busy person tonight, Chris. The next one's for you as well. Totally down your side. Would not the introduction of habitats for bats on farms help reduce the mosquito population? Wagtails and other birds, dragonflies, insects, especially mosquitoes, thought ought to be given to natural predation of mosquitoes by bats and birds.
Well, firstly, to say that bats need all the help they can get. They're really struggling in the UK and Northern Europe. So I would encourage introduction of habitats and boxes and things that bats can use. Unfortunately, we don't have any evidence that increasing natural predation of midges, bats do eat midges, we know that much, but we don't have any evidence that that would control the population. We are talking about a huge biomass of insects that bite animals. Thousands of insects are biting animals every night, and birds and bats would just, they just can't cope with those numbers. So natural predation is not going to be a way of resolving this. But yeah, please do put back boxes and let's treat our wildlife as best as possible.
Then the next one: why were only animals with severe symptoms euthanized in the Netherlands rather than all those showing symptoms? Wouldn't this have increased the spread of disease?
I can't speak for the Dutch and their policies, but I would imagine that the spread of the virus was so quickly they were playing catch up. So once this virus basically was detected, within a few weeks it had spread over a substantial part of the country. So by the time that kind of machination could get going, we were seeing disease everywhere. And so then, as Jose was saying, the implications of locking down, of culling, become vast when even the testing capacity of the country involved. So it may just be a logistics challenge. If you can get to those first signs, first animals really quickly, so if we had an incursion and we identified an animal really quickly, an infected farm, then culling may actually prevent or at least slow transmission. But I think in the Netherlands that horse had bolted before they could actually get on top of it.
So I think, yeah, I was going to mention on that, Chris, that also you did mention, and we know that some of these animals would have mild clinical signs and they would recover on treatment, and so the indiscriminate destruction of these animals, it sort of adds insult to injury to farmers that perhaps can salvage some of these animals and they become immune to the disease as well if they overcome it.
Yeah. I think I'll also add a tiny bit here. Indeed, in the Netherlands, and I had some conversations with the people there as well. At the beginning, they didn't euthanize, and indeed, first it's government who cull, and then it continues on. And at the beginning, they didn't euthanize a lot of the animals. They tried every single thing in the book to try and save them. At the latter end of this outbreak, they actually were a lot quicker to euthanize animals. So I haven't had any but there were parameters that they followed. And if those parameters ticked certain boxes, they would actually euthanize them quicker to save them, to go through the pain that they'd have. And so, yeah, it's difficult. But I'm assuming with blood samples, you can find out what stage animals are. Right. So I have another question for you, Chris. How long does an animal remain infectious to midges after it has been infected itself?
Okay, so as soon as we can detect viremia, essentially those animals start to become infectious. So that's day 3 or day 4, and the infectivity to midges increases till you get to the peak viremia sometime between day 5 and day 10. Once we get to day 14, that infectivity decreases. So once we get to day 20, it's really difficult to infect midges from these animals. It's not an absolute, but it's a sharp decline. So I'd say that infectivity of ruminants that are infected to midges declines substantially between 14 and 20 days. But we can infect individual midges. So if we can infect, say, 100 midges at peak viremia, by 20 days, we're infecting 1 or 2. So it's that sharp drop off, and that's probably due to the interactions with the immune response of those animals.
Thank you. So then here's another question. When a confirmed infected animal recovers, if clinical signs, or is allowed to remain on farm, does it act as a reservoir, or can it even abort next season? Interesting.
So this comes back to what you're talking about, Sasha. Actually, kind of, if these animals recover, then they will be immune to Bluetongue virus. They've been naturally vaccinated, and so would remain as immune livestock and part of the herd immunity. So once that infectious period has passed, once we get past 20 days or so of infection, those animals don't present a significant risk of virus transmission to vectors, and so provided they are recovering well and there are no welfare issues associated with them, then actually that's great because they've become naturally immune to BTV3 or any other virus.
On the second part, could they abort next season, Chris?
I don't think so, because that the abortions occur due to the impact of infection during a specific period of pregnancy. So outside of that sort of viremic period, the following season, those animals should undergo a normal pregnancy. If there is damage that's unknown in that animal, then that might incur fertility issues in the future. But I'm not aware of that being a particular thing about Bluetongue.
Yeah, I suppose it also depends on what stage of the pregnancy they became ill, and if it's a long pregnancy, then I think recently in Holland they've had a cow abort with, but maybe they hadn't been ill, in all fairness, but they had an 8-month-old calf. But anyway, thank you. Right. So there's also I'm just going through all of them. So there's a question about whether any positive entire rams and do I'm in a room here. Do positive entire rams present the same risk for maintaining virus in the testes as bulls do? I'm not sure if you're able to answer that one, Chris, and if not, we can always come back to this one at a later stage.
Yes, I'm not aware of that. I mean, it's still, even in cattle, it's a really understudied area because of the nature of infecting bulls and things. So yeah, I don't know with rams. We'll have to come back to that one.
Yeah, we'll ask the other specialists, Carrie amongst others, to get an answer, and we'll come back on that one. Then, are electric fly killers a solution to control midges?
Unfortunately not. They work really well in delicatessens and things. You've seen kind of flies go to them and get zapped. Just again, the population density of these insects, they will get in, and you run the risk of, if you have an attractive trap, you actually run the risk of increasing the attraction of that area to midges, and not all of them will go to the trap. You'll collect a lot of midges, but you will not necessarily impact virus transmission or even biting rates on the animals, unfortunately.
Fabulous, thanks. Then there was a question at the beginning, were these drugs used separately or in combination, and are there any recommended combinations there? Now, obviously, Jose was talking about the Netherlands at that point in time, and it's obviously specific to what happened there, and we could have a different situation here. So I think it's very much a case by case situation, and we obviously have to follow our own cascade. But I will can go back, or Jose can go back to the Netherlands and check if they did a combination or not, or just used different drug choices.
Yeah.
And Jose, do you have anything else to add?
No. I was I was just going to say that we can perhaps ask if there was any particular combination they used. So and come back.
Yeah.
Yeah.
That's fine. I think that's all the questions that have come in. I think, wait 2 seconds, I think something has just come in again. Right, I think that's it. Thank you all for your attendance here, and thank you for your questions. I think this has been really helpful, and I hope we will not come across the disease or the level of disease they had in Holland, because I think that was a very testing time for all, animal, human, everything. And but I hope that this has a sort of hopefully it'll help you when you're out there in the field and you come across this and your clients do have cases. A lot has been tried in Holland, and I mean, they really tried the weirdest things, it's obvious this is a pretty horrible virus, and it is a virus, there's not really a lot more than supportive therapy that we can do. So I hopefully we won't see it anytime soon, but thank you all. Thank you, Jose, for your presentation. Really appreciated. And Chris, thank you as well for all your knowledge, and it was really helpful. Thank you for everyone else for having joined us, and we will see you again at some point. Good evening.
Thank you very much, everyone.